Hello students! Alright, today we continue with 11.4 and we're going to be looking at both animal fertilization and human fertilization. So the essential understanding is pretty vague, and this is something that you've probably been studying since earlier years. And it's that fertilization in animals can be internal or external. So we know the difference, right? We know that external fertilization involves the fusion of the gametes, the sperm and egg, outside the body of a parent. It is more common in aquatic animals because the water acts as a medium for the gametes to travel. But this makes the process a little bit more susceptible to other influences like predators and pH changes. Therefore, because many of these gametes might get lost to these influences, that means that species that undergo external fertilization are going to release large quantities of gametes to compensate for losses. The process of releasing these gametes is called spawning. And this is a good video here that shows you mass spawning by coral. You might want to look at on your own time. Internal fertilization involves the fusion of gametes and egg and sperm inside the body of a parent. That means that one gamete needs to be introduced inside the body of another parent, and that is called copulation. Terrestrial animals typically use internal fertilization because you don't want the gametes to desiccate or dry out if you expose them to those conditions. And internal fertilization is a way to protect these gametes and these embryos, but it has a cost to the parent because, of course, the parent has to carry that embryo / fetus inside the body and then give birth to it. Here is a very nice video, and I am going to play this, that shows you internal fertilization, the fusion of an egg and sperm. And I just want you to pay attention and see how quickly that happens. And so there's the sperm that just came in, that went through the barrier of the zona pellucida. And now notice how the zona pellucida hardens. And notice how the two nuclei here are going to fuse. And then notice how quickly the cell starts to divide. So there's a time lapse right there. So an hour has passed. So an hour after fertilization, you already start seeing mitotic division. So it's pretty quick, pretty interesting to see. So let's look at human fertilization in a little bit more detail. So the essential understanding is that fertilization involves mechanisms that prevent polyspermy. That means that you don't want more than one sperm fertilizing one egg. So there are three key processes that we are going to focus on. One is capacitation, two is acrosome reaction, and three is cortical reaction. So let's look at capacitation. So capacitation occurs after ejaculation. That's when the male deposits its sperm inside the female. And you see that chemicals released within the uterus of the female are going to dissolve the sperm's cholesterol coat. So this is what the sperm looks like before capacitation, and this is what the uterine chemicals do to the sperm. And when that cholesterol dissolves, that means that the sperm has higher motility and can therefore reach the egg. Also, the acrosome cap destabilizes because the acrosome is what's going to penetrate the egg, and then it needs to release those enzymes to burrow through that jelly coat or zona pellucida. All right, then we see the acrosome reaction here, and this is when the sperm actually reaches the egg, and the sperm has to dissolve that jelly coat within the egg. The sperm has to dissolve that zona pellucida. So the sperm is going to push through the corona radiata, which are the follicular cells, and then the acrosome, which is the head of the sperm, is going to kind of bind here with the zona pellucida, and then the enzymes from the head are going to be released. So those enzymes are going to digest the egg jelly coat of the zona pellucida, which is a glycoprotein matrix. And then the sperm is just going to push its way through, and notice that the nucleus, which is part of the acrosome head, that is going to go inside the egg after the sperm kind of has docked with the egg membrane, and the sperm nucleus will enter the egg, along with the centrioles also that are found within the sperm. So after that happens, there needs to be a cortical reaction to prevent other sperm from binding. So upon fertilization, you see here the sperm, and you only see one sperm, right, actually binding and docking here with the membrane proteins of the egg. The enzymes in the acrosome destroy the glycoprotein matrix of the zona pellucida, and the nucleus is going to be able to come inside the nucleus. But once that happens also, besides the enzyme destroying that jelly coat, right, that's going to allow for this sperm to enter, once that head or that nucleus enters, those enzymes are also going to harden the glycoprotein matrix of the jelly coat. So that's going to prevent other sperm from attaching. So notice that within that membrane of the egg, you have these kind of like docking proteins. So once this zona pellucida or this jelly coat has hardened, those sperm binding proteins just disappear, right? So the zona pellucida hardens, and another sperm will not be able to penetrate. So therefore, you are preventing that polyspermy, or more than one sperm fertilizing one egg. Okay, so that was a quick video. I hope you enjoyed it.